Good morning. Can you hear me?
Yes.
Perfect. Oh my. How about now? Can you hear me?
Yep.
Still not doing it. All right, 1 second. It's technical difficulties here. Okay, now can you hear me?
Yes.
Perfect. Now my headphones are working. Okay. I don't know why it's not letting me host as the normal grad student government person, but we're doing it. It's
It works. Whatever.
Yep, whatever works. So let me get the slides up. Session 3. Don't. Laura, I'm glad you're here. Do you know why it's not letting me host as grad student government and only as pro dev?
What do you mean you're not? Which account did you use to sign up?
My pro dev one, but it shouldn't matter. I'm co-host for GSG as both.
I know I had the same issue, just doing it from the GSG account.
So should I do it from my personal account then?
No, it wouldn't let me join with that one either. Use the GSG at MTU one.
Oh, I don't have the login for that.
Yeah, you do. I can send it to you. You have access to that. Do you want me? Or I could wait, sign in that way and make you host. I'm already in as that person. Do you want me to do that?
I made you co-host. That means I can leave and come back if you send me the login. But I should be a co-host. I mean, it says it's recording, so it's probably fine, right?
No, that's an automatic thing. Give me 1 second.
This is weird because I got the email saying I was put on as a co-host for my pro dev account. Sorry guys, having some technical difficulties with the logins.
Now it won't let me. What are you having issues with?
Like it, so when Renate logged in, his said graduate student government as his name, so it was under the right email.
Mm-hmm.
When I opened the Zoom meeting, because I used my ProDev email, it opened it under my ProDev. So the recording is going to go to my ProDev account instead of the GSG account.
I think it goes to both.
Okay, then I'm not going to worry about it.
Okay, and otherwise you can, like, we can talk about sharing links later.
Oh God, it's never easy. Yep. Okay. Thank you.
I'm here.
I'm glad. It's a struggle.
Just to make you feel better, I am on the GSG and I'm seeing that the recording is being recorded under GSG, so no worries.
Perfect. Okay. Cool. 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 That's great. Okay. So we're waiting for one more judge. How is everyone doing today? Excited to be here?
Doing great. How are you doing this morning?
I'm good. It's certainly a time trying to coordinate everything for this event, so it's something. But happy to be here. Happy to see everybody who is here. It's gonna be a good time.
I have one quick question.
Sure.
We will be able to advance our slides on our own, correct?
Yes, I'm gonna go over the rules first thing once everyone is here. I'm going to be giving you remote control, crossing our fingers that that works. If it doesn't, or if you prefer it, you can have me advance the slides by giving me a prompt saying next or proceed or whatever. But hopefully, yes. And I can actually give you that right now so you can practice since you're the first one up if you'd like. Cool, that seems to work. Oh, you're muted.
Yeah, I was just gonna say it looks like it's working.
Cool. Perfect. I'm going to take that away from you.
And then I had a video on my slide. Well, the sound should work for that, correct?
Hope so.
Okay.
It should. Everything should work just like a normal presentation. Um, but being on Zoom, I'm not gonna make any promises.
Okay, because I know like when I was practicing last night and when I did the share screen, I had to do like there's an option at the bottom to optimize or play video over Zoom.
Okay, let's go to your slides right now, shall we? Oh,
Yes, it was like 28 or something was the one that the video was on.
okay, you just stop me when I get there.
Sorry, Isaac, showing all your results. Okay, there it is.
I can hear it.
I can't hear it.
You need to,
Oh.
whenever you share screen, there's going to be two little boxes underneath. You need to click both of them. So you have to stop your sharing and start again.
Oh my God.
Sorry.
What? Wait, say that again, Laura. Sorry, the video
Stop
was
your
starting.
stop sharing your screen.
Okay.
Start again. Before you select the screen you want to share, there are two little boxes at the bottom of that thingy.
Oh, optimize for video clip
Both,
and share sound.
both, yeah.
Got it. Okay.
Okay,
Good.
Now we're
it should
ready.
work now.
Try that one more time. Um,
Community transmission
hear
are
it?
high throughout the entire U.S., with Michigan reporting record numbers of cases and hospitalization.
I taught online for a year. I I know all the things.
Could all of you hear it?
Yeah.
Okay.
Yep.
Great.
Thank you.
You are welcome. Thank you for making sure I had that right. Because I would not have known.
Sorry, I have a lot of animations to work through here.
No, you're good. Now I can't see anyone, so this is interesting. Show me the participants. I guess it doesn't super matter. Okay, I'm gonna go ahead and get started. We've got everyone here as far as I can tell. I wonder if it.
You have a little screen on top of your presentation.
Where?
Center of the screen, you have a blocked version. You have something in there that you're seeing.
Do you see it now?
Nope, offset.
Okay, thank you for bearing with me, everyone. All right, so we're gonna go ahead and begin. Thank you all for coming out and taking part in this year's Graduate Research Colloquium. My name is Margaret, and I'm going to be your moderator today. Just a couple of ground rules before we get started. Each contestant will have 12 minutes to present. Your time will start the second that you start speaking. I'll give you a 1-minute warning by interrupting your talk over Zoom, so I'll just say 1 minute remaining. As soon as the last speaker finishes, Q&A. Okay. As soon as the last, after the talk, we'll go into Q&A with the judges. As soon as the last speaker finishes the Q&A session, you'll be given the remote control access, and you need to accept this access for you to control the screen. If you don't want the remote control, you can just inform me, and in that case, you can give me a verbal cue to start and say next and proceed, and we can go through the slides like that. I will be introducing you before each talk, and then you can jump directly into your presentation to save time. At the end of the presentation, judges can unmute themselves and ask questions to the participants for about 3 minutes. While the judges are scoring, I will then moderate questions from the general audience, but I ask that the audience does stay muted during the presentation and they can drop their questions into the chat box. So with that, I'm going to go ahead and give Isaac remote control here. So our first presenter is Isaac Wedig, presenting a practical application of blood flow restriction exercise. You can go ahead and proceed.
Okay, there we go. All right, well, thank you for the introduction and good morning, everyone. So blood flow restriction, or BFR, consists of performing exercise with mechanical compression applied to the upper portion of a limb. And this is usually done using an inflatable cuff. And this inflatable cuff compresses the blood vessels within the limb. And this serves to reduce the amount of blood flow going to the working muscle during exercise and limits most of the blood flow from leaving the muscle during exercise. So the major benefit of performing exercise with blood flow restriction is that increases in muscle size and strength can be achieved similar to that of traditional resistance exercise, but while utilizing much lighter weights. And so accordingly, blood flow restriction offers an alternative way to increase muscle size and strength for individuals that cannot participate in heavy traditional resistance exercise. And this includes populations such as the elderly, those with orthopedic limitations, and also individuals with various disease states. So in order to standardize the degree of blood flow restriction during exercise, the pressure within the inflatable cuff is based off of something called arterial occlusion pressure, or AOP. And this is the minimum amount of pressure that is required within the cuff to completely stop blood flow through the limb. And once this value is obtained, then the pressure within the cuff during exercise can be set as a percentage of that value to ensure that blood flow during exercise is only reduced and it's not completely restricted. So implementing blood flow restriction safely and effectively requires some specialized equipment. So first, you have to have a device to restrict blood flow, or a way to actually compress the limb. And typically this requires using an inflatable cuff system with a pressure sensor. And then secondly, you have to have equipment to assess arterial occlusion pressure. And so this requires you to have Doppler ultrasound, or a way to directly assess when blood flow through the limb has been completely stopped by the restrictive device. And so collectively, the high costs associated with this equipment needed to implement blood flow restriction severely limit the access of this type of exercise to physical therapists, to coaches, or anyone who could benefit from utilizing this type of exercise. So in order to make blood flow restriction more accessible, previous researchers have tried to remove the need for direct measurement of arterial occlusion pressure by developing ways to estimate this value using an individual's blood pressure, their leg size, and their leg composition, or the amounts of muscle and fat that they have present within their leg. So each of these variables have been shown to be predictors of arterial occlusion pressure and have been used to create prediction equations that can estimate that value for an individual. The only issue is these prediction equations have been created for use with high-cost specialized cuffs, and prediction equations have not been created for more affordable options. And thus, the purpose of this project was to develop a practical way to implement blood flow restriction in the lower body, utilizing a low-cost thigh blood pressure cuff as a restrictive device, and then generating a prediction equation to estimate arterial occlusion pressure for this specific cuff, using an individual's blood pressure, their leg size, and their leg composition. And so specifically, my aims with this project were to, slides are a little delayed here. First, to determine whether blood pressure, leg size, and leg composition are predictors of arterial occlusion pressure with this type of cuff. And then secondly, to determine if leg size or leg composition measures are better at predicting this value. And then lastly, to create two separate prediction equations, one utilizing measures of leg size and one utilizing measures of leg composition, to estimate arterial occlusion pressure with this specific cuff. So moving on to the methods. So 69 healthy adults with normal blood pressure were recruited, and 23 females and 46 males were included. Individuals attended the laboratory for one visit where measures of blood pressure, leg size, and leg composition were assessed. And for blood pressure, both the systolic and diastolic blood pressures were taken in the arm. For leg size, thigh circumference was measured using a tape measure. And for leg composition, skin fold thickness was taken on the front of the thigh using a caliper, and this measure served as a predictor of the amount of fat within the limb. And muscle cross-sectional area was estimated using both the thigh circumference measures combined with the skin fold thickness, and that served as an estimate for the amount of muscle tissue present in the limb. And then lastly, arterial occlusion pressure was assessed using the thigh blood pressure cuff and Doppler ultrasound. So in order to determine which variables were predictors of arterial occlusion pressure, 2 multiple linear regression models were constructed. And the first model utilized measures of leg size and blood pressure to predict the outcome variable of arterial occlusion pressure. And the second model utilized measures of leg composition and blood pressure as predictors. So after running the analysis with all the respective predictor variables in each model, the analysis was then rerun, including only the significant predictors. And then those simplified models could be compared to see if leg size or leg composition were better at estimating arterial occlusion pressure. And then also those simplified models were used to generate prediction equations to estimate that value for each of those measures. On to the results. So here is a table summarizing the leg size model. So thigh circumference and systolic blood pressure were significant predictors. And the model explained 37.7% of the variance in arterial occlusion pressure and had a mean prediction error of plus or minus 9.73 millimeters of mercury. Utilizing only the significant variables from that model, this simplified model still explained 37.7% of the variance in arterial occlusion pressure, and the mean prediction error increased slightly to 9.81 millimeters of mercury. And then this simplified model was used to generate the following prediction equation to estimate arterial occlusion pressure. Here is a table that summarizes the leg composition model. So muscle cross-sectional area, skin fold thickness, and systolic blood pressure were all significant predictors. And the model explained 36.2% of the variance in arterial occlusion pressure and had a mean prediction error of 9.78 millimeters of mercury. Again, taking only those significant variables, this simplified leg composition model explained 37% of the variance in arterial occlusion pressure and had a mean prediction error of 9.79 millimeters of mercury. And then this model was used to generate the following prediction equation using the leg composition measures. So in summary, I found that within a model accounting for leg size, thigh circumference and systolic blood pressure were significant predictors of arterial occlusion pressure, again, in the lower body when utilizing a thigh blood pressure cuff as a restrictive device. And a model accounting for leg composition, muscle cross-sectional area, skin fold thickness, and systolic blood pressure were significant. Importantly, a model accounting for leg size explained similar amounts of variability in arterial occlusion pressure as a model accounting for leg composition. And so this suggests that leg composition may not need to be taken into account when estimating this value, and that a simpler model utilizing just thigh circumference and systolic blood pressure can provide just as good of an estimate. And this is consistent with previous literature that has shown that thigh circumference and systolic blood pressure are predictors of arterial occlusion pressure when utilizing more high-cost specialized cuffs. The only difference, Lenicky and colleagues explained more total variance in arterial occlusion pressure, 49% versus 38% in my data. And this may be likely attributed to the difference in the width of cuffs used. So I utilized an 18 centimeter wide cuff, and Lenicky and colleagues utilized a 5 centimeter wide cuff. And so the width of the cuff may be impacting the relationship between these predictor variables and arterial occlusion pressure. Also importantly, this is the first study to find that field-based estimates of leg composition can serve as predictors of arterial occlusion pressure. So previous work has utilized more direct lab-based measures of leg composition, but have found very similar results that these measures are significant predictors and that they also explain similar amounts of variability to a model just accounting for leg size. So in conclusion, a prediction equation using field-based measures of leg size and blood pressure can serve as a practical way to estimate arterial occlusion pressure in the lower body. And importantly, a cost-effective cuff paired with a prediction equation to estimate this value provides a very low-cost way to implement blood flow restriction without the need for specialized equipment. And also, a prediction equation that utilizes very easily obtainable measures such as thigh circumference and systolic blood pressure can be easily employed by physical therapists,
1 minute remaining,
clinicians, and also coaches. And so ultimately, this practical approach to implementing blood flow restriction can help to make this type of exercise more accessible to be used as a way to increase individuals' muscle size and strength for those who cannot engage in heavy resistance training. So thank you very much for listening, and be happy to take questions now.
awesome. Thank you, Isaac. Now I would like to ask judges to unmute themselves to ask questions, please.
I'm gonna jump in so we can get this moving along. Great presentation, Isaac. Thank you so much. I have 3 very short related questions. So with the blood occlusion, the arterial occlusion, if I understand correctly, the blood that's gonna pool in the lower limbs is gonna be the blood from the veins, correct?
It would be, I'm sorry, what was the last part of that?
So because what you're occluding is the arterial blood flow, the blood that would pool in the lower limbs is actually from the veins, correct?
Yes, correct. Yep. So it's going to stop. So, I mean, during exercise, you're only restricting arterial blood flow. So you still do have blood flow into the muscle. And then obviously the veins have a lower pressure. So they're going to be a little bit more, I guess, not fully occluded. So yeah, you will get, an accumulation of blood and metabolites down below the cuff when it's placed there. So yeah, generally that will be the fluid that would be accumulating there.
Okay, so that does lead to sort of lower oxygen in the muscles, which is the effect that you get with the exercise as well. But that also leads to the production of, the lactic acid and all the byproducts from that. So at a time when you run these experiments, how long were they run, or how long can you occlude this, before you think it's unsafe?
Yeah, and that's a big topic of concern with blood flow restriction is the safety. It seems, a little dangerous to limit blood flow. And I always go back and look at like the tourniquet literature. In surgery, you are applying tourniquets, and in surgery, you're fully occluding blood flow to the limb. And the cuff can stay on for, 2 to 3 hours during surgery. And so, important to look at when you're exercising, there still is blood flow. And there's good evidence too that during exercise, because you have the muscles actually contracting, you have a muscle pump that is actually helping to move blood in and out of the muscle. So the safety part of things is there are certain populations where you need to be more careful doing blood flow restriction exercise. An individual with hypertension who may, have a dysregulated ability to sort of, regulate their blood pressure during exercise to begin with. They may have, a very heightened hemodynamic response and cardiovascular response. For the most part, though, during the exercise interventions, the cuff is not staying inflated for more than 10 to 20 minutes at max. And then the cuff is deflated after that. And with the amount of literature that's been published on it and utilizing it so far, there's been very small, very limited reports of negative consequences of that. It is painful, it doesn't feel comfortable when you're doing it, but a lot of the things that are happening that are uncomfortable, the accumulation of the metabolites, that all seems to be part of the mechanism of why it works. And so that's kind of what you're looking for with the exercise. So hopefully that kind of helps answer your question.
Absolutely. And you also answered my last question about the population that you would exclude from such an exercise. So thank you so much.
Yeah, you're welcome. Good question.
Thank you. And with that, we're going to have to move on because of time constraints. So our next contestant is Ashley Hawke. from kinesiology and integrative physiology, which, sorry, Isaac was also from KIP. And she will be presenting on building a healthier information environment during COVID-19. So I'm going to go ahead and give you remote control now, and your time will start as soon as you start speaking.
All right. Thank you for that introduction. Oh, sorry. Oh my goodness, I am so sorry.
You're good. There should be arrows at the bottom, and you should also be able to use the arrows on your keyboard.
Okay, sorry. So sorry about that.
You're good.
All right. So over the past 2 years, our understanding of COVID-19 has unfolded in real time and continues to evolve daily. With so much new information coming out, it can be difficult to keep up on what is current and accurate versus old information. We've also seen an influx of health misinformation, which is information that's false, inaccurate, or misleading. And this has actually led the World Health Organization to classify this launch of misinformation as an infodemic. Some consequences of this infodemic include people rejecting public health measures like wearing a mask and social distancing, refusing to get the COVID-19 vaccine, or using unproven treatments to treat COVID-19. Sometimes misinformation is shared intentionally and or maliciously, but many people who share are often just voicing concerns or seeking questions to answers they may have, and aren't sharing to intentionally misinform. Regardless, sharing misinformation is literally costly. Since May 2021, misinformation and disinformation are estimated to have cost the US between $50 and $300 million worth of harm each day. Because of this, the US Surgeon General, Dr. Murthy, has called on all Americans, including educators and universities, to do our part in helping build a healthy information environment in our communities, workplaces, schools, on social media, and even in our homes. In response to this action call, my fellow graduate students and I are doing our part to provide reliable COVID-19 information to the Michigan Tech campus and surrounding communities. We've come up with a few ways to do this. The first being developing and distributing a bi-monthly COVID-19 newsletter to our department. This newsletter was reviewed by Dr. Kelly Kam, who is an assistant professor and epidemiologist in the KIP department. In the newsletter, we highlight the current case trends in Michigan and Houghton County, share a relevant research study of the week, share a healthy living behavior tip of the week, and then strategies on how to find correct information, focusing on reliable websites that have .gov, .edu, or .org in them. We also link useful resources such as the CDC, Johns Hopkins, and the Western UP Health Department, as well as miscellaneous information for students, such as upcoming vaccine clinics, testing locations, and current travel guidelines. On this slide is an example of what the brief looks like. This one actually was sent out to the department in December before winter break. Additionally, we recently published a letter to the editor titled Building a Healthier Information Environment During COVID-19. Oh my goodness, I'm so sorry. As graduate students, we've also participated in the monthly COVID-19 virtual town hall series sponsored by the Health Research Institute. On the town halls, we've provided commentary on various topics such as COVID misinformation and the importance of physical activity for physical and mental health. The goal of the Town Hall series is to increase awareness about COVID-19 and how it impacts health and society, especially our surrounding community. We've also recently published a letter to the editor titled Building a Healthier Information Environment During COVID-19 In the journal Advances in Physiology Education, this letter outlines what we as graduate students at Michigan Tech and graduate students at other universities have done to relay useful health information to the campus and surrounding communities. We've also updated the KIP department website to include COVID-19 resources, such as where to get tested on or off campus, what to do if you're exposed or test positive for the virus, resources for mental and physical health, and again, the weekly COVID briefs that were previously mentioned for those who may not be on the email list to receive them. And then lastly, we created a short 2-minute video that reiterates the continued severity of the virus, ways to keep yourself and your community safe, and once again, what to do if you're exposed or test positive. Thanks to the Center for Rural Health at Northern Michigan University, this video is now playing on local TV stations and in movie theaters across the UP. The video was actually created back in December at the start of the Omicron surge, but is still relevant today as we are still in the midst of the pandemic. So with all of these modalities, we've been able to broaden our impact and reach more audiences, such as fellow researchers, students, and then those who reside in Houghton and all over the Upper Peninsula. So here is a shortened 1-minute version of the video.
In 2020, COVID-19 was the third leading cause of death in the United States and continues to be a
I'm not sure why this isn't working. But again, it reiterates the continued severity of the virus and what to do if you test positive. So I'm just gonna.
In 2020, COVID-19.
Okay. So why does this matter? Why is combating misinformation important? First is that by combating misinformation and learning the best, most efficient and impactful way to get important health information to the public, we can then use those tools to address other important health issues, such as risk factors for metabolic disease, importance of physical activity, or types of environmental related illnesses, and potentially find new ways to relay health information to the public. Additionally, scientists have already stated that unfortunately, this won't be the last pandemic we see in our lifetimes, and that our generation will see more in the future. As graduate students, we're preparing to become future leaders and will likely be in leadership roles by the time the next pandemic comes around. So again, by learning the most efficient way to relay important health information to the public and reduce the amount of misinformation that's in circulation, we may be able to, quote unquote, shorten the length of future pandemics. And then lastly, we're seeing a lot of misinformation being shared on social media. So by continuing to combat misinformation, we can hopefully help bring awareness to the type of information that people are consuming, as well as the source, and continue suggesting getting information from anything or about anything, not just COVID-19, from credible sources versus from Facebook. So I want to end with a quick take-home message on how we can each do our part to build a healthier information environment. First would be to rely on information from credible sources and those with health credentials, such as public health experts, your primary care provider, and again, websites that end in .gov.edu or .org. Second would be use social media with caution. There are some legitimate sources of information like the Copper Country Strong and State of Michigan Facebook pages, but do think twice and do a quick fact check before hitting share. And then lastly, talk with your family and friends about where they get their health information from. If they say a website that maybe isn't the best, kindly suggest credible and reliable sources that they can check out instead. Promoting a healthier information environment has been a team effort, so I wanted to give a special thank you to all of those who have been involved, and I can take any questions that anyone may have.
You still have 2.5 minutes, so do you want me to go back and try to see if I can get that video to work?
Yeah, if we could, please. You can take me off remote, whatever.
Well, because I'm pretty sure I can get it to work when it's not shared screen.
COVID-19 was the third leading cause of death in the United States and continues to be a leading cause in 2020.
Can you guys hear that?
Yes.
Oh, perfect.
Over 850,000 people in the US have passed away due to COVID-19. Levels of community transmission are high throughout the entire US, with Michigan reporting record numbers of cases and hospitalizations.
Some strategies to keep yourself and community safe during the fight against COVID-19 are: get vaccinated, get a booster shot if you're already vaccinated, wear a mask, wash your hands, watch your distance from others, and walk to stay physically active.
It's also important to get tested regularly, to quarantine if you are a close contact, and isolate from others if you test positive. With all of this information, we are asking everyone to do their part to keep ourselves and our communities safe. Make sure that you're getting your information from credible sources.
Awesome.
Thank you for playing that again.
Yeah, no problem. I'm sorry that didn't work for whatever reason. But now I'd like to ask the judges to please ask any questions that they have.
Can you hear me?
You are very quiet.
Okay, let me try to increase the.
That's much better.
Okay, so very good presentation, I would say, and it's very important, especially in front part when we have like unlimited information. So, and you have like reached in all areas, especially through video and right through publication and stuff. But mostly like we face problem with our like individuals in our own family. Like they said they also have their own source of information. So I was thinking, do you plan on working on that area? Like when we know like from the scientific facts, we have our understanding, but they have a lot of information out there and they have their own perception based on that. So how do you interact with them and convince those peoples who are like very dear to you? You don't want to just sideline them. So is there any techniques you are developing or something which can help individuals who want to help with their family member?
So that's a very good question, and one that I get asked a lot. So we found that especially now, a lot of people are unfortunately set in their ways. And like you said, we don't want to just sideline them. So the best strategy, and this is one that I've actually implemented with my own family, is just laying out all of the credible sources like CDC, non-biased news sources, so that they have those resources and try to get them to come, I guess, to those conclusions on their own. Like they don't want to feel pressured into changing their minds. And another thing is to just ask people why they chose that viewpoint, and just have a conversation with them, I guess, is the easiest way. Like you don't want to attack them, you don't want to berate them. And again, you just want to make sure that they're coming to that conclusion on their own so that they don't feel, I guess, forced or coerced into a different line of thinking that may be different than what they're used to.
Do you think like your group or like you should work on this area as well, like develop some method which can help people to some kind of guidelines, just like you have information in this one?
Yeah, that's something that we could definitely look forward or look to do in the future, because we're always looking for ways to continue this work because it's so important. We could come up with just a general guidelines, but that's I guess, kind of difficult because so many people, everyone is different in their own ways. And so it's hard to come up with kind of an overarching guideline, but we could definitely set up and distribute, and that could be something that we put on future briefs or on our KIP website of just ways to approach someone who may have, I guess, differing viewpoints.
Thank you.
Is there time for another quick question?
If it's very quick, yes.
All right, depends on the answer. Was there a reason on what type of media you used to spread this information? Was there any indication of why one of them would be more suitable than other?
So another good question. We were just trying to hit, I guess, all possible audiences that we could. So for our briefs, we actually surveyed KIP staff and staff, faculty, and students on how they would like to receive this information. So that's how we came up with the brief. And then our video was actually ended up being kind of like a capstone project for the end of the semester, and the Center for Rural Health actually reached out to us and asked to use that to hit more of those audiences who may not have as much access to the internet, maybe those older audiences that maybe don't use email or social media regularly. So trying to kind of hit it from all fronts.
Thank you.
Well, thank you, Ashley. Dr. Rao and Dr. Bashir, are you both able to see the judging form right now with session 3 and see all of the people that you're supposed to be filling out comments for?
Yes.
Yes, we can.
Awesome, thank you. All right, well, with that, I'm going to go ahead and proceed then. Next up, we have Amanda Studinger from chemistry presenting on energetic competition in the complex affinity of paracetamol with water and oxalic acid. I'm going to go ahead and share the remote control with you, Amanda, and you can begin when you are ready. Is there a problem?
Oh, she's muted.
Oh, okay, that's why I can't hear anything. Amanda, can you unmute yourself, please, and tell me what's going on?
All right, can you hear me now?
Yes. Now I'm going
Awesome.
to go
I
ahead.
completely lost my controls for muting and chat.
Sure,
And
no problem.
so
That's what happens. It's a little bit funny. Okay, so can you still control my screen? Yep.
Yep,
Looks good.
perfect.
All right, when you're ready, I'll start the timer when you start speaking.
All right, thank you. All right, so we're looking at the energetic competition of complexation affinity for paracetamol with water and oxalic acid. So just a brief outline of the talk. I'll go over some of the background, talking about intermolecular interactions, and specifically about APIs, active pharmaceutical ingredients, paracetamol itself, and then the method that we're using, density functional theory, to do our study. We'll go over the motivations and objectives for this, the methods, and then we'll go over some results and conclusions. So briefly, intermolecular interactions. And specifically for this work, we're looking at hydrogen bonds. So intermolecular interactions are those attractive forces between molecules. And this is a reversible binding, meaning it isn't like a covalent bond where you're actually making a molecule, but it's an interaction that can be really strong, and it plays a big part in our everyday lives. So it's reversible binding, and there are different types of reversible binding that we see. We see electrostatic, hydrogen bonds, London dispersion, dipole-dipole, and ion dipole. So these molecules here just kind of give you a brief idea of the different types of interactions. So if you see in the lower corner, there you see some blue and red around a molecule, and this is an amino acid. And what it's showing is the electron density, the amount of time that the electrons spend over one part of the molecule over the other, and how that isn't balanced. So when the electrons are spending one time on one side of a molecule over another, it's not balanced. We see electrostatic charges, like you would have electrostatic balloon over your carpet or something, or you can see ionic charges. So here with this amino acid, we have a Zwitter ions. We actually see ions where something's actually lost an electron or gained an electron. Over on the right-hand side, you see a phospholipid, and these are commonly used to make up our cell membranes. And you see these long tails that don't have any color, and that's what we would describe as that London dispersion force. And then you see colored ends, that's the polar end of our, what makes up of our cells. So these interactions are extremely important, especially when we're talking about in our own bodies, but they happen everywhere. Specifically for this project, we're going to be looking at hydrogen bonds. So looking at hydrogen bonds, there's a hydrogen bond donor, which is a part of a molecule that is electron deficient, meaning that it has a slightly positive charge to it, and a hydrogen bond acceptor, so something that is electron rich. So talking about active pharmaceutical ingredients, abbreviated API, it's the active drug molecule. So oftentimes when you have a drug, it's been mixed with other things to make it into its pill form or to make it less concentrated so that we can actually safely ingest it or have it injected into us. So when we're talking about the active drug molecule, that is the specific drug that is interacting in our bodies. They can be naturally occurring or synthesized. And when they are synthesized, they're manufactured, dried, encapsulated, or pelletized. And we run into certain barriers when we're synthesizing based off of some of these interactions. So there's a well-known case about a crystal that was being formed for an HIV drug that worked really, really well. And then all of a sudden it stopped working for people. And they didn't know why it wasn't working. What turned out was that a different crystal structure was being made, and it was so stable that it was going through everyone's digestive systems and not actually being absorbed. So looking at intermolecular interactions is really important, not only in the manufacturing process, but also what happens in the bodies and then afterwards when the drug's not being used anymore. So we're specifically looking at co-crystal conformers. So that is an API with another molecule. It can create stable crystal structures, and what we're hoping for is the desired physiochemical property, right? So can you ingest it? Can you absorb it? Can you use it, metabolize it? Does it bind to what we need it to bind? And then can we get rid of it later? So we're talking about the pharmacokinetics, the pharmacodynamics, and then later when the drug isn't being used, or if there's excess drug or excess materials from the manufacturing process, what are the environmental impacts? So waste processing, transport, storage, and uptake. There's an interesting story for this area in the UP about meth labs and people getting raided with meth labs, and they put the materials that they make down the drain before they get raided by the police. And then our wastewater treatment plants are seeing a lot of these materials in their water, and they're not necessarily prepared for getting rid of these materials in their wastewater. So understanding how these materials interact with the environment and how they interact with waste processing will help us down the road. So I'm looking at a specific co-crystal with paracetamol, which many of you might know as aspirin, paracetamol and oxalic acid. So this is a solid structure, and it creates this co-crystal that makes these flat sheets, so coplanar sheets, and then they stack on top of each other. So there's this interlayer stacking, and it's held together by hydrogen bonds in the sheets, and then pi-pi interactions for the sheets interacting, stacking together. This particular co-crystal has unique hydrogen bonding sites. So oxalic acid itself, which is the smaller molecule, has both a hydrogen bond donor and a hydrogen bond acceptor. And then paracetamol here is also a hydrogen bond donor and a hydrogen bond acceptor. So we're going to look to see at every different site how it behaves. So to do this, we're using computational chemistry, a theoretical method of modeling what the energies would be for this crystal. Using electron density instead of the wave function, it's called density functional theory. Let's see if I can get my equations to show up. So some of you might be familiar with what's called the Schrodinger equation, which was a way to describe the kinetic and potential energy of a system, looking at atoms or molecules and figuring out what their wave functions were for their electrons. From there, you can figure out what the total energy is of that system. So the Schrodinger equation is a great model, but you can't use it for anything other than hydrogen, because once you have more than one electron involved, it makes it really difficult to solve. So there are different methods, different ways to get around that, and one of them is density functional theory. All of these are approximations, and they're just ways to better describe it, and not anything exact. So instead of using the Schrodinger equation, you use the Cohen-Sham equation, which got a little messed up, but that's okay. It looks almost identical to the Schrodinger equation, except we're using different values for the kinetic and potential energy and for the actual wave function. So using the Cohen-Sham formula for based off of electron density to find the wave function, and then, and then the Cohen-Sham Hamiltonian, which is a way that we describe the kinetic and potential energy. So the rotation, the movement of the molecules and the atoms and electrons, and the attractive forces that we see on also the potential energy that we see. So one of the biggest buggers with computational chemistry is that effective potential. That's the potential energy, and being able to really accurately predict or describe what electrons are doing and how they interact with each other, and how does that affect the energy. And so that'll be the big player with our work here. So motivations and objectives. I had a timer for time, I'm running low. So we use the density functional theory to look at the intermolecular interactions. We're trying to better understand drug design and manufacturing and see if we can model and accurately model these interactions. So looking at methods, we used multiple different functionals for our calculations. Because each functional has a different way of describing what our system might look like, we wanted to use multiple functionals and see how they behaved in our different system, but we wanted to keep it the same. So our wave function, the way we describe the electrons, we kept that the same across the board, and we used different functionals or different ways to describe the kinetic and potential energy. We looked at different complexes, PCA and oxalic acid, and we looked at the unique hydrogen bonding sites. You can see those are numbered 1, 2, 3, and 4, and then 1, 2 with oxalic acid. We looked at each site, and we looked at PCA and oxalic acid together, PCA and water, and oxalic acid and water. And then we looked at what it looks like in vacuum with nothing around it, and then what it looks like what's called implicit solvation, where we surround it with a charge that would be the equivalent of water. So there are a lot of results, and I'll try to go through these quickly. Basically, we looked at the geometries and we looked at the energies. So in the background is this gray area where after we did our work, you can see that the geometry changes when we try to optimize and try to calculate what the energy is. And the colored part is what the crystal originally looked like. And you can see, especially here at position 4, there's a big difference in what the computer program finished off and said the geometry would look like when it's optimized versus what it looks like in the crystal. So we wanted to see how those changes in geometry affected the overall energy and stability of the cool crystal itself. This is just showing at position 4, there was a lot of variation. You can see how much it's twisted around. So we looked at hydrogen bond
We have
distances.
1 minute remaining.
Thank you. To see how that changed, and we looked at angles. And basically we concluded that the crystal itself, the co-crystal conformer, was really strained. It was scrunched together and there was some strain at different positions. And these results are just showing the changes and how when it was in a crystal versus when it was able to relax, how it changed based off of that. And then the error bar showing how much of a difference there was between each method that we tried. We looked at configuration energy, so bringing these, what's the difference in energy based off of the geometry. And again, we saw big changes at position 4. BSOC correction is just the computer itself. Sometimes the different parts overlap, and so you over approximate energy. So we wanted to take that out of consideration by calculating how much energy that was. And complexation energy. Oh, all right.
Oh, I'm going to take back control here. Oops, sorry. And one of our judges was lost during that time. So it's all right. It's okay. But yes, for now, I will open the floor to questions for Amanda. Oh, you had such a nice animation.
I know, it's all right.
Dr. Rao, are you back in now? Hopefully soon.
All right, I am back. Thank you.
Okay. I'm sorry about that. It's one of those things when someone else has control of my screen, I can't admit anyone.
That's okay. I didn't think about it. I'm so sorry.
No, you're totally fine. Now I will open up for a couple of questions from the judges for Amanda, if there are any. Go ahead.
So I can go first.
Please do.
So I know you could not explain your results, so I will just ask, like, what did you learn so far from this project, and how it can help in this industry?
Yeah, so it took too long, but that last animation slide really showed that overall, when oxalic acid was the hydrogen bond donor, or if you were in a polar environment like water, that the hydrogen bonds were a lot stronger. And those were the pieces that really dominated or influenced how the crystal came together. So what we saw were 2 positions specifically that really were the influencers in how the crystal came together, and the other positions didn't really matter. So why does that matter? Because there are a lot of different sites that can make hydrogen bonds, but when you're designing drugs, it's good to know if there's one particular site that's really, really strong, and that's going to influence everything and how it's going to bind to the receptor that you need in the body, then that's really all you need to focus on. So instead of worrying so much about all the different sites or tweaking all the different sites, maybe you can tweak just one site in the molecule, and that makes a big difference, especially with synthesis.
Yeah. Thank you.
Is there time for another question?
Yep.
Thank you, Amanda. My question is just about, I know you mentioned what are some of the hard things to work on this, specifically, the computational modeling. But for you specifically with this project, what was the biggest challenge that you have to overcome, or is there something that you foresee will be a huge challenge for you?
Really for us, the most time consuming, but the best thing that we did was testing all those different functionals and how the different functionals behaved and how they interacted, and did it allow the computation to complete. So when we first started this project, we were trying a completely different basis set and trying a completely different method almost, and we were not getting results. We weren't getting anything to to finalize. We weren't getting any of our calculations to work. So the biggest challenge is really finding the right methods and the right basis set that will allow for results and then hopefully allow for accurate results.
Very good. Thank you so much.
Okay, for time's sake, I'm going to continue now. I apologize. We are going to go over time. But if everyone is ready, I'm going to invite Arslan Amer to talk. Arslan is from Biological Sciences and we'll be talking about exploring alternative approaches for pancreatic cancer therapy by targeting cysteine metabolism. So Arslan, I'll give you remote access and I will start your timer when you're ready.
Okay, let me just.
I can barely hear you.
Sorry, just making sure I do not have the remote access right now. Oh, it's
There
a
you
bit
do.
slow, I guess.
Yeah, it's a little bit delayed, but yep, I'll start your timer when you start talking.
Okay. So hello, everyone. I will be talking about an alternative approach for pancreatic cancer therapy by targeting cysteine metabolism. First of all, regarding what is the importance of the pancreatic cancer, it might not be as high in the incidence as, let's say, lung cancer or the breast cancer, because those guys have a really high incidence rate. But for mortality, pancreatic cancer is the fourth most lethal cancer right now in the world, and it is suggested to rise to the second position by 2030. The major reason for that is that it has a very poor prognosis. It's very aggressive. And also there are very difficult treatment options for this. Majorly, the only treatment right now is surgical resection and chemotherapy. And even with that, resurgence rate is very high. So only 6 people out of 100 survive this. So there's a need for a therapy which can be used, which can kill cancer cells specifically, because right now the only therapy options we have are a little bit general. One of the things when we talk about cancers is their high growth. Cancer cells they proliferate and grow very fast. And there are a couple of changes involved with them, like metastasis, where they change their form so they can go and invade different organs. And also they need supply of blood for them to invade other things. So all of these things require a certain bit of metabolic changes. These metabolic changes help the cancer cells to better adapt and then progress. So while targeting the metabolic changes is a good therapy option for us, it has been done before. There's a very common effect known as Warburg effect, where they figured out that the cancer cells are highly dependent on sugar because on glucose because they are developing so fast. So if you kind of limit the supply of glucose, the cancer cells die off while the normal cells retain because they have their function, they can maintain that very, very well. So similar approach has been associated to cysteine also. Cysteine is an amino acid, and while cysteine has a lot of functions in the cancer cells specifically, it's basically involved with the GPX system, which is a lipid metabolism, lipid peroxidation cleanup mechanism. So when the cells grow too fast, they will have a lot of metabolites which they're running through, and one of them is the reactive oxygen species. And reactive oxygen species can affect lipids, and high lipid oxidation causes a cell death called as ferroptosis. And the major cleanup mechanism of this is through a GPX4 pathway, and glutathione is one thing which is a substrate to GPX4. So while we block cysteine, we cannot get enough glutathione, and this system cannot function, leading to cell death. So while this mechanism occurs in cancer cells, in normal cells, because lipid peroxidation levels are low, they do not die off. But however, when you block cysteine in a cancer cells, they do die. So this has been reported in breast cancer. This has been reported in colorectal cancers. So what we aimed was to check it in pancreatic cancer and to see how well it works. And if not, then how can we make it work even better? So in the pancreatic cancers, when we do check, this approach. However, we figured out that some of the cancer cells were very much responding to this therapy. As you can see here, when we block cysteine, this is a cysteine blocker, the cancer cells die off. However, there were certain cancer cells which were resistant, and they were not responding to this therapy option. And we wanted to figure out why this was happening. So for our models, we used 6 cell lines, and 2 of them which were super sensitive, and 4 of them which appeared resistant to these blockers. And we wanted to figure out the genetic basis or different therapeutic options to make them more sensitive. So the first idea was to check what kind of genes are upregulated or downregulated in these 2 groups. Interestingly, we found selenoprotein genes to be highly differential in these. Selenoproteins are proteins which are majorly associated with selenium, and they have usually 1 single selenium atom in their active site. And they're majorly enzymes which are used for antioxidant pathways, which makes sense. Interestingly, if GPX4, which we talked about, is also a selenoprotein, so that makes sense. And also it involves TRX pathway, which is also involved in redox signaling. However, the one gene which we did found very high was SEP1, and SEP1 is majorly a transporter protein or a storage protein for selenium. It is the only protein which has 7 to 18 selenium residues instead of just 1. And it's basically an indicator of how well the cells are harboring selenium or using selenium. So that's why they will make more SEP1 in order to make it more accessible. So this leads us to that maybe selenium itself is very important, and because of GPX4, it makes sense. So first thing we wanted to check was whether the selenium was involved. So selenium is very important in translation of these proteins. So if the selenium will not be there, these proteins will not get translated and they get stopped. So just a brief kind of thing is that the protein will get truncated if there is no selenium there. So when we added the selenium to the system, we figured out that it did rescue the cells from cysteine deprivation. So if you see, this was cysteine deprivation, the cells were dying, but as we added more selenite or selenium to the system, the cells started getting rescued. And this actually shows that the selenium is important for cysteine dependence therapy for pancreatic cancer. But why it was happening was a little bit unclear. Best hypothesis for that is GPX4 itself, because we know GPX4 is a selenoprotein and it is working. So we tried to check whether the GPX4 was upregulated, whether its activity was good. And if you can see, when the selenite was applied, GPX4 was, its expression jumped, even on messenger RNA level a little bit, but majorly on translational level, because selenite is majorly affecting these proteins at translational levels. So that makes a little bit of a simple story that, okay, your SEP1 was up, and when you give selenite, your selenoprotein gets high, which is a cleanup mechanism. However, this does not relate to cysteine that much, because you see, when we say that we have given selenite and it is upregulating GPX4, still it does not have any effect on the cysteine pathway, because cysteine deprivation is causing glutathione to go down. So even though the enzyme is going up, you don't have the substrate which is needed for the function. So unless selenium is also affecting somehow glutathione levels, this upregulation of GPX4 is not a very good explanation of why selenite is rescuing the cells, because if the substrate is not there, enzyme will not function. So when we check the value of the glutathione, it got depleted, if you see here. So when we add Arslan or selenium depletion, glutathione goes down. Interestingly, in selenite treatment, it didn't go back up. It was still down. It was even a little bit more down than usual, which makes sense. If you're upregulating the pathways, the glutathione would be used up more because you're upregulating the enzymes. So this created this problem that how is this happening? There is no glutathione, yet selenite is protecting the cells. And for that, we did a literature search and we want to figure out what happened. So as I mentioned earlier, TRX is also a pathway, and it's also a selenoprotein pathway, and it works redundant to GPX pathway. So if you see here, GPX and TRX perform essentially the same function, which is cleaning up ROS. However, and they start with the same NADP as like a hydrogen donor, but they work redundantly to each other. And because both of them have selenium proteins in them, so it can be hypothesized that the selenium will affect both of these systems equally. So what we basically did was we employed blockers to block each of these pathways out. And when we block the GPX pathway, which is majorly associated with ferroptosis and cysteine depletion, the rescue by the selenite was not affected at all. So that was interesting. And when we block the TRX pathway, however, if you see here, the Arslan plus the selenium rescue of the cells was abolished. And this actually kind of confirmed that in these systems, selenite is affecting the TRX system much, much more than the GPX system, which is traditionally seen as the major driver for ferroptosis. So this also begs the question that, oh, if you block the TRX pathway in the resistant cell lines, would they become sensitive as well? Because that's their major form of resistance coming from because of high selenium and high SEP1. So we tried basically a synergistic effect where we added cysteine and TRXR blockers together. In those resistant cell lines, which we talked about earlier, we said that they were not responding to this therapy, and they did. So if you see by just Arslan alone, most of them were resistant to this and they were not dying off. But when we added the TRXR blockers, you will see all of that resistance goes down and the cells die because of this. So this led to this whole therapy notion that while pancreatic cancer is affected by cysteine depletion, however, certain cancer cells will not be affected that much because of a high TRX pathway and high selenium content. So if we can block selenium during this therapy, or we can block the TRX system, which is ultimately the downstream of it, we can make the Arslan or cysteine depletion therapy a much more better therapy to be used in the pancreatic cancer system. And also SEP1 or selenium content can be used as a marker that if you see that there's a high selenium content, you can employ this strategy of TRX blockers. Otherwise,
1 minute remaining.
Otherwise, without it, just the Arslan system is good. So that's basically what the summary is for what I have said. So co-treatment of cysteine and TRX blocker can produce a synergistic effect and can be used as a therapy for pancreatic cancer. And with that, I'm good. For acknowledgement, my lab and my Fulbright is my sponsor. And thank you very much.
Awesome. Thank you, Arslan. Now I would like to ask the judges to unmute and ask any questions they have.
Dr. Bashir, do you want to go first again?
No, I don't think I have any question for this one.
All right. I do have a quick question, Arslan. Very good presentation. Lots of work here. So you did mention that, with similar to Warburg effect, there is a increased uptake of glucose, and limiting that glucose has metabolic effects, and the cells could potentially undergo cell death. But cancer cells also have a lot of capability to adapt to their metabolic environment. So can you comment on the adaptation with changes in cysteine and either short-term or long-term impact of that?
So one thing which I would comment on, like when we talk about the TRX system, I think that is one of the adaptations, because when cysteine was majorly a driver force for the GPX system, and that's, I think they compensated something that they went to the TRX system. And similar things can happen in the future, because if the cells, you try to give them this therapy and the cells have resistance, they try to balance it out with some other system. One thing for now is that both of these systems are dependent on selenium. So that's one part, that blocking selenium can definitely abolish both of these systems. But however, there are other redox cleaning systems already in the cells. It's very complicated. So yes, they can effectively go there. But one thing I will say that the TRX blockers, when we employed it in our system with Arslan, their killing mechanism was very fast. So it's like within 8 hours, which was even Arslan alone requires 24 hours or something. But this was very fast. So one thing which I would say that if you kill them fast enough, and if this does not affect the normal cell at all, then there might be a chance that you basically limit that. But of course, there's always a chance of clonal expansion. The clones which are resistant will then grow out, and I think more research should be done, and we can just try to modulate all of these things, because there's a lot of mechanisms that are still unknown. So that is something that will come up when you employ one strategy or the other. But yeah, that's a problem that definitely is.
All right, great answer. I do have a follow-up, which was sort of linked to what you said as well. You did mention that the killing is very rapid. Do you have any data that indicates how the cell death is occurring? Is it stress initiated? Is it apoptosis? Do you know what the mechanism for the death is?
Yeah, when we checked, I mean, it's still, I will not finalize anything, but it is majorly necrosis, necroptotic pathway, because what we feel like is because both of these systems, when blocked, the lipid peroxidation goes to such a high level that it actually starts like cell death very quickly. We have some data where we have employed certain blockers for known necroptotic and ferroptotic pathways. In those data, we have seen some cell lines have a little bit of different pathway, but general trend is, of course, it's necropoptosis, but different kind of necropoptosis, which makes sense because maybe there are some underlying mechanisms going on which try to protect it from one, so it shifts to the other. But majorly it is necrosis, and yeah, it's like cell injury, high lipid peroxidation. Yes.
Thank you so much.
Okay, well, with that, I'm going to stop the recording. Thank you everyone for coming. There is a poster session tonight from 5 to 7 in the ballroom. I encourage you all to attend. Yeah, so thank you very much. Judges, you can stay on to confirm with each other if you'd like, but other than that,